Okay, so I think this is where we left off yesterday. Beginning of 13.4. This is where we're at. We just have one section left. Is that correct? All right, so we had left off comparing and contrasting mitosis versus meiosis. So let's finish this up here. Let's talk about the origin of variation. Genetic variation originally starts with mutations, which remember are changes in an organism's DNA. So so that's how we get different forms of a gene, is from mutation. Mutations create different versions of a gene. We call those alleles. So that when we're talking about a trait, like when I talked about pea plants and purple flowers versus white flowers, why we have the pea plant has a purple flower gene and a white flower gene is because of a mutation. There's a change in the DNA, and so they make different proteins and therefore give different traits. And so mutations not always are a bad thing. So sometimes DNA can mutate and it can be a bad thing. A lot of our genetic diseases are caused by mutations because a gene mutates and you don't make the right protein, and therefore whatever job that protein was supposed to do in your body is not happening, and those symptoms as a result of not having those proteins give you a specific genetic disease. But some mutations can be good and they can be random, and so some mutations can cause a different protein that can be made that might be beneficial, and so on and so forth. So mutations are the original source of genetic diversity and why we have different versions of genes and therefore can have different combinations of genes, and so on. And so it's the reshuffling of these versions of genes during sexual reproduction, where you make your egg and your sperm cell and they unite and you make your children, that produces the genetic variation. So we're going to look at three sources of genetic variation. So the behavior of chromosomes during meiosis and fertilization is responsible for most of the genetic variation that arises in each generation in the kids. So the three mechanisms we'll look at: first is independent assortment of chromosomes, second is crossing over, which you've already talked about, but we're going to look at it with genes, and the third is random fertilization. So independent assortment. So homologous pairs of chromosomes orient randomly at metaphase 1 of meiosis. In independent assortment, each pair of chromosomes sorts maternal and paternal homologs, those are homologous chromosomes, into daughter cells independently of the other pairs. The number of combinations possible when chromosomes assort independently into gametes is 2 to the nth power, where n is the haploid number. So for humans, our haploid number is 23. For humans, n is 23. And so there are more than 8 million, or 2 to the 23rd power, possible combinations of chromosomes, and therefore combinations of genes that we can get in our egg and our sperm cells. So just based upon how the chromosomes line up. And I want to illustrate this independent assortment with this picture here. So this picture shows you two possibilities. So here's possibility number 1, and here's possibility number 2. And so let's look at this. So possibility number 1. So we have our pairs of chromosomes here. They're lined up in metaphase with their partners, and we have two pairs of chromosomes here. So this is one pair, so this is one replicated chromosome, the second replicated chromosome, and this is the other pair. And they color code them to show that one came from mom and one came from dad, all right? And that's why we have pairs in the first place. So during meiosis, remember, we separate the homologous chromosomes, so one long and one short chromosome is going to go into this cell, one long and one short chromosome is going to go into this cell, and so that's meiosis 1. Meiosis 2, the purpose of meiosis 2 is to separate the sister chromatids. So that's what we see here. And so we have one of every kind of chromosome here. And so let's go through an example with genes. And so let's do the red and the purple and white flower gene. So let's say big R is purple. Purple is dominant to white. Little r is is white. So we're going to put some genes on these chromosomes to see how combinations of genes can change. So that's one trait. So that's trait number 1. Let's look at another trait. Trait number 2. Let's do, well, this is a pea plant, so that would be quite an organism. So let's do another trait with peas is whether the peas are round, so the outside is smooth, or sometimes the peas can look wrinkled, so they're kind of like shriveled up. So it's round versus wrinkled. So I'll use ends. All right, so this is a round pea, and a little end would be a wrinkled pea. So these are my 2 traits that we're looking at. And we're going to assign these traits onto our chromosomes. And so remember that when we look at a chromosome, that this piece of DNA is an exact copy of this piece of DNA held together at the centromere. And so remember, genes are segments of DNA. Right, guys? Segments of DNA. And so what happens is that these guys are homologous pairs of chromosomes. So this blue chromosome over here, let's say right at this spot right here on this piece of DNA is the segment of DNA that codes for making either the purple or the white gene. And so what we're going to do is make this pea plant that's going to go through and make gametes and go through meiosis. We're going to make them heterozygous for this trait. What does the word heterozygous mean?
It's got 2 different traits. Well, not 2 different traits. It's got a recessive and a dominant.
As a recessive and a dominant. So let's make them heterozygous for this the flower color. So they're going to be big R, little r. This is the plant's, is that the plant's genotype or phenotype? That's the plant's genotype. It's the gene combination they have for that particular trait. So what that tells you is they have a purple gene and a white gene. What color are they going to appear? They're going to appear purple because purple is dominant. So when this plant is going to go through meiosis, we have in this spot here a big R. This right here, what's going to be on the second chromosome in that exact same spot? Is it going to be a big R or a little r? People always get confused with this. Think about the relationship between this piece of DNA and this piece of DNA. It's identical, right? So this is just a copy of this. So on this one, I don't have enough room, so I'm gonna put the big R on top there. So there's a big R in that same spot for that, in that piece of DNA for a flower color gene. Then this one, in that same spot, there's also a gene and that second chromosome for flower color, except this time it's not the big R, it's the little r, right? So therefore, it's gonna be a little r, and this one also has a little r in there. Okay, let's make this second trait on the other pair of chromosomes. So let's say in the DNA here, right here is the trait that's going to determine whether or not they're round or wrinkled. And so let's make them heterozygous as well. So therefore, let's say this is going to be a big N, so then this chromatid is also going to have a big N. This one would have a little n, and this one would also have a little n because they're exact copies. So therefore, we have heterozygous for both traits, but the traits are on different chromosomes. Question?
So do we look at this similarly to how we look at Punnett square?
Absolutely. That's what we're going to get into. Yeah, absolutely. That's the connection I wanted you to make. Right. So what happens here is these homologous chromosomes separate. So what you're going to have is the chromosome with the big R's here, and this red chromosome with the little r's here, and the one with the big N's here, and the one with the little n's here. So these two, what combination are they going to have? Big R and a big N. So they're going to have a big R and a big N in these two, and these two are going to have little r, little n. And notice it says combination 1 and 2. Okay, so that's that. Notice here, possibility 2, what did they do in possibility 2? They flipped. If you look here, these two flipped. all right? And so, because every time a pair of chromosomes comes to line up down the center of the cell, they have two different ways that each one of them can line up. Just like if I had two people come up, like say I have a boy and a girl and stand against the board, sometimes the boy would be on the left and the girl would be on the right, or they could do it either way if I didn't tell you how to line up. The same thing with these chromosomes. So there's two different ways that these chromosomes can line up. So if you look here, and so this is the big R chromosome here and the little r here, and this was the, this we're going to switch, so this is the little n, and here the big N. So now we have the big R chromosome separated into this cell, the little r chromosome separated into this cell, but now the little n chromosome is in with the big R chromosome in this cell, and the big N here is with the little r. This is a different combination than we got here, so therefore at the end we get another combination here. So we can get combination 3 here, big R with a little n, or we can get a little r with a big N. Just based upon how they line up. Follow me? All right, so then going back to the last couple bullets, it said there's a formula, and it's 2 to the nth power. So in this case here, if they only have 2 pairs of chromosomes, what's this cell's haploid number? Let's write this. So this is a diploid cell. We would say 2n equals what? 4, right? So therefore the haploid number is 2, right? We have a long chromosome and a short chromosome, right? Haploid number is 2. So 2 to the n power, 2 to the 2nd power, this is the only possible combinations. There's 4 possible combinations for this. Now imagine though, we don't have 2 pairs of chromosomes. How many pairs of chromosomes do we have?
23.
We have 23 pairs of chromosomes. So imagine the 23 pairs, each chromosome pair can line up in one of 2 different ways. And how chromosome pair number 1 lines up is different and has no relationship to how chromosome pair number 2 lines up, and 3, and so on. So because there's 2 different ways for every chromosome pair, that's where the 2 comes, all right? And then there's 23 pairs lining up. It's 2 to the 23rd power. So if you put in your calculator 2 to the 23rd power, that is over 8 million possibilities. So what that means is every time a male or female makes an egg or a sperm cell, you have only a 1 in 8 million chance of ever making the same egg or sperm cell with the same combinations of genes, just based upon how the chromosomes line up. All right? All right? So that's just with how they line up, and that's called independent assortment, the way that they line up independently of one another. Okay? Following me? Question? It looks like you have a question. No?
This marker smells like butter.
Oh.
He's just...
Okay, you're just sniffing the markers. Okay. All right. So,
Sorry.
no wonder you looked a little confused. All right. That's thing number 1. That's the first thing that creates genetic variation. Let's look at the second thing is crossing over, which we hopefully are familiar with. Crossing over produces recombinant chromosomes, which combine genes inherited for each parent. Crossing over begins very early in prophase, as homologous chromosomes pair up gene by gene. The homologous portions of the 2 non-sister chromatids trade places. This is all a review. Crossing over contributes to that genetic variation by combining DNA from 2 parents into a single chromosome. And so this is what I want to show you here, is how you can get new combinations. And so I'm going to show you this by assigning genes on the chromosomes like we just did with the independent assortment. So in this case, notice there they only show you 1 pair of chromosomes. So on this pair of chromosomes, you know, they're made out of DNA, and we're going to look at 3 different genes on there. So we're going to look at 3 genes on the same chromosome. So let's say right here is a gene for, and it doesn't really even matter what trait it's for. Let's say that they're also heterozygous. So we can do the same with the purple and the white. So we have a big R, and this one would be a little r because they're heterozygous. And then let's do towards the middle, like right above the centromere here. Let's do another trait. What letter should I use? Let's use B's. Let's make them big B and little b. And then let's do towards the bottom here. Let's do big N, little n. So they're heterozygous for 3 different genes, whatever those genes are. So, so now, in showing crossing over, color coding is very, very helpful. So you can see the actual exchange of genetic information. So if you have like color pencils or something, I have some color pencils up here if you'd like to do some color coding. I think it's easier to see, but if you think you got it, you know. So what's happening here? Notice that these 2 chromosomes in prophase are doing a double crossover. They're going to cross over and exchange the end piece of DNA and the end piece of DNA. So therefore, this outside chromatid right here is going to have still a big R, a big B, and a big N. That outside chromatid is not involved in crossing over at all, okay? This part here, that inside chromatid, is going to be involved in crossing over here. And so therefore, it is going to have a big R, but the big R is it's kind of hard to see. It's behind there. It's going to switch spots, all right, and be on the red chromosome. And we'll see that when they separate here. And then, is the B affected in this crossover at all? No. And so therefore, we're still going to have, I'm going to squeeze that in here, a big B there. It's not affected at all. And I try to draw the N so that they would be affected. And so the N, the little n, is going to, the big N is going to eventually be on the red chromosome. So we have the big N is kind of underneath here. It's hard to see here. Then this chromosome, the red chromosome, the outside chromatid is not involved in crossing over. So that's still going to have the little r, the little b, and the little n. That does not affect it at all. But what is going to be affected is the little r here. So this little r is going to actually be put on this chromosome here. Doesn't affect the B, all right, so that's still a little b here. You have to draw it kind of small to fit in here, so this is still a little b. And then it will affect the little n. The little n is going to go onto the blue chromosome. So they drew this again. I don't know necessarily we need to put all the genes on here. They're just showing you the only difference between here and here is what's attached to it. I'm trying to show you that the spindles are attached to it and they're going to be pulled to opposite sides of the cell and be split into 2 different cells. So this would be the same as this. So then let's look at when they separate into 2 different cells. What I'd like you to do is follow this through and put the genes on these 2 chromosomes and on these 2 chromosomes in terms of what you'd end up with at the end. So the chromosome, the blue one here, going to the left here. So the left chromatid is still going to have all the same original genes on it. So you should have the big R, the big B, and the big N on here. But it crossed over 2 times, and so therefore instead of a big R here, now it has what was on the red chromosome, which is the little r. This is still going to be a big B because it wasn't involved in the crossover, and this one here, instead of a big N, it now has the piece of the red chromosome, so it's going to have a little n. So that's what that one looks like. Then,
It's just, no, it's the projector, it's the fan.
It literally sounds like either engine or
Keeps getting higher and higher.
It just went away from me. But it might have just been so high.
I still hear it.
You're still here.
Yeah, it's getting higher and higher. I still hear it. No, it wasn't.
Sounds like it's
Is Claire charging us there?
Mr. Davenport on the other side did the same thing. He's like searching his room. That's Mr. Claver doing some kind of experiment.
Oh my god.
I was curious.
We should stop momentarily. All right, as long as we know what it is. All right. So this red chromosome here, the outside chromatid also. All right, guys, pull it together. Is not affected, so it's going to still have all the recessives. So little r, little b, little n. And then it's going to also here, because it's switched with the blue, have the big R. Still the little b on the left-hand side because that wasn't involved in crossing over, and having the little n. And so then what you're going to have here is, if this is the egg cell that is given to the kids, you're going to have a big R that parent's going to pass on all dominant genes. If this is the egg cell, they're going to give a little r, a big B, and a little n. This one, they're going to give a big R, little b, and little n. And this one, little r, all recessives. And these guys right here are called recombinant chromosomes because the only way that you can get that combination of genes is by the DNA recombining, crossing over. All right, so these guys are not possible unless crossing over occurs. So that's the recombinant chromosomes there. And so crossing over is a huge input, guys, of genetic variation. And we have no way to actually measure how often it occurs. So therefore, remember that we have 23 pairs of chromosomes. All these 23 pairs of chromosomes can do crossing over. They can do it in random spots. They can do it once, twice. And how they cross over one time when you're making your egg and sperm cells are different than the way that they're going to cross over the next time, and so on and so forth. So imagine this, okay? So in addition to just the way your 23 pairs of chromosomes line up, you have over 8 million. You have a 1 in 8 million chance of ever making the egg or sperm cell the same way twice, just based upon how they lined up. Now you throw in crossing over, that throws that number way up. We don't know what that number is, but it's way more than 1 in 8 million. And so therefore, taking into account the independent assortment in addition to crossing over, creates huge varieties of egg or sperm cells, which creates genetic diversity. All right, and so that's why, two parents can have 8 kids, and each of those 8 kids are very, very different, have a different a combination of genes that are very different from one another, and it's due to these things, okay? And so the last thing has to do with fertilization. So random fertilization. fertilization adds to genetic variation because any sperm confused with any ovum, an ovum is the unfertilized egg. So the fusion of gametes produces a zygote with any of about 64 trillion diploid combinations. And they got this by multiplying that statistic, 1 in 8 million possible combinations for the egg with 1 in 8 million possible combinations for the sperm. Crossing over adds even more variation, and each zygote has a unique genetic identity. So, so what does this mean, and why are we multiplying this? So imagine this. So let's say, even though you only have 1 in 8 million chance, so let's say the father of the couple makes the same sperm cell twice. So there's 1 in 8 million chance of that happening. The mother makes the same egg cell twice, so that's 1 in 8 million chance. The chance that the mother is going to release that particular egg during that month, and then that particular 1 in 8 million sperm is going to fertilize that egg. That's the random fertilization, and so the chance of that happening, what the rule of probability is, the rule of multiplication is you take the individual probabilities of two things happening. So 1 in 8 million chance that dad's going to make the same sperm cell, 1 in 8 million chance that mom's going to make the same egg cell. The chance of those occurring together, you multiply them together. So 1 in 8 million times 1 in 8 million gives you 1 in 64 trillion chances that that would even happen. And so therefore, it's very unlikely that that's going to happen. And that doesn't even throw in crossing over. So then you throw in crossing over, and the likelihood that you're ever going to get two kids, two people on Earth that are genetically identical is next to nothing, basically. I mean, it's possible. It's still possible, but it's very, very low probability. No. Twins are different. I'll just address that real quick. Twins, identical twins, one sperm fertilizes one egg. You get a zygote. They start dividing and to develop into an embryo. What happens in identical twins is the massive cells separate and then continue to divide as two different people. And that's why they're identical. Yeah. And fraternal twins, non-identical twins, is the mother releases that month 2 egg cells. She has 2 egg cells, 2 different sperm cells fertilize those egg cells and then grow up into 2 babies. So fraternal twins are like, genetically, they're no different than a brother or sister or 2 sisters that are born at different times. They're just born at the same time. Okay. So, so therefore, so we are very unlikely to have, similar people, all right, or identical people. And that's important. Why is it important to have this much genetic diversity for a species?
Isn't it for like self-controlled diseases? So like certain things aren't spread to your offspring. It's like I can't figure like, but if you had everyone be the same, there's more possibility for more mutations and stuff like that.
So if everybody's the same, there's more chance of, how would there be more chance of mutation?
Because everyone's so like if I got the exact same thing from my father and then like from my grandfather and everything on, I'm gonna have like I'm not gonna become immune to everything new. Or well like I don't know how to word it. But
I think maybe you're maybe talking about the fact that we can have like dominant and recessive, like, I don't know.
no like I don't know, like I can't explain it.
It has to do with kind of
Saying that you don't
adaptations.
evolve.
We wouldn't evolve,
Yeah.
all right, wouldn't be able to adapt, Nathan?
So that like if one natural disaster happens, it doesn't wipe out the entire species.
Yeah.
I remember the easy example of like there's an animal and one variation runs fast and one doesn't because of forest fire. The slow one will die, but the fast one will live on. So the species survive.
That's exactly right, all right? And so having diversity helps with survival, that's right. I just didn't make the connection there, all right? So, so absolutely, so this last little bit here, let's finish this up. That's called natural selection, what Nathan just described. Results in the accumulation of genetic variations favored by the environment. Sexual reproduction contributes to the genetic variation in the population, which ultimately results from mutations. But the genetic variation makes it so that at least if the environment changes hopefully you have some individuals that have some kinds of traits that help with the survival, and therefore while not everybody in the population might not survive, some will, and therefore keep the population of species alive.
I've heard this somewhere, I'm not sure this is exactly how I heard it, but I've heard that in like incest, I know that when a baby is, like when a lineage of incest begins, the gene pool becomes smaller and smaller because the same genes are being used over and over again, and that can cause mutations as well.
It doesn't cause mutations. It doesn't necessarily cause mutations, but what happens with people who are related is that, like, you can be, like a lot of genetic diseases are recessive, so you could be big R, little r, and the big R is the normal gene where you don't have the disease. So I could be big R, little r for a particular disease. So I'm carrying the disease gene, but I don't show it physically because it's a recessive gene. But what happens is if you have children with people who are closely related to you, there's a higher likelihood that they're also have that same gene in their, versus somebody who's unrelated that possibly is not even in their family at all. So therefore, when you have kids, you have this likelihood that the kid gets those two recessive genes that come together, and so then kids can have a lot more problems because of that.
That's what I was trying to say.
Okay, that's
I
what you're,
figured that's
okay,
what he was
okay, gotcha. All right, so yes, that's what I meant by dominant, when I said I think you're talking about dominant recessive and hiding genes, that's what okay, so I was kind of getting you.
More on what like she was talking about. I know one argument against like GMOs, them being all like genetically similar, is that if there's like a disease that were to wipe out a bunch of crops, then the more of them that were similar, the less of them that would survive. That's an example of survival without biodiversity.
That's correct, absolutely. All right, how do we get out of here? Okay, so we are done.